So, what we've taught so far as it relates to metabolism. Most bacteria will respire aerobically when oxygen is present. This applies to obligate aerobes, right? Obviously, that's the only way they're respiring. Facultative anaerobes, as we've talked about, will utilize oxygen in the electron transport chain when it's available because it's going to produce the largest amount of energy. But as the name implies, facultative anaerobes, they can respire anaerobically, usually due to fermentation when there isn't any oxygen. Microaerophiles, again, require a small amount of oxygen. They will obtain a small amount of energy from glycolysis, but they will capture a relatively large amount, as we calculated out before, right around the 30s, if they can give those electrons to the electron transport chain, do oxidative phosphorylation, because that oxygen is accepting the electrons at the end of that transport. And we're generating, remember, lots of ATP, lots of energy. The main role of the Krebs cycle in energy production is to feed a lot of those reduced coenzymes, those electron carriers, to that electron transport chain. They recycle their reduced coenzymes to oxidized, right? They lose the electrons by giving them to, as I said, oxygen, and that forms water. So we're breaking down, we're removing electrons, we're oxidizing sugars like glucose, and the carbon is given off in the form of carbon dioxide. The mainly bacteria can resort to fermentation when oxygen is absent, right? So when there isn't any oxygen, they're usually going to resort to fermentation. This applies to our facultative anaerobes, right? When there's no oxygen, they're usually doing fermentation, right? That's how they're growing anaerobically without oxygen. Aerotolerant anaerobes, for the most part, are just doing fermentation, right? They don't utilize an electron transport chain, whether aerobically or anaerobically. And then there are some organisms that are obligate anaerobes. They rely on fermentation, or they can do a form of anaerobic respiration, where they're using electron transport chain, but oxygen is not the final electron acceptor, right? There's some other molecule that's accepting those electrons. So they capture a relatively small amount of glucose, it's free energy, strictly from substrate level phosphorylation and glycolysis. So remember, some ATP is generated in glycolysis, but not a lot. Not as much as you get from getting the electrons from the Krebs cycle and going to the electron transport chain. They recycle the electron carriers that have gained electrons, those reduced coenzymes, by adding them back to pyruvate, forming the different acids that we talked about last time and tested for, alcohols, and gases. And they ultimately are breaking down glucose, right? They're oxidizing, they're still removing electrons, but not generating, not completely breaking it down. They're still ending up with 1 to 4 carbon molecules, right? These acids, these alcohols, and then also gas production. So you may have noticed something peculiar, right? Most of the bacteria will respire aerobically when oxygen is present. That's what we said, right? Many bacteria can resort to fermentation when oxygen is absent. No problem, right? So under certain circumstances, some bacteria, though, will they'll ferment the carbohydrate even when oxygen is present. So oxygen is present, right? You can get a whole lot of energy, but instead they're fermenting. Why would they do that? You have a question, Andrew? Oh, you figured out what you think your organism is? What you think? Nope. We'll go over your results after. Okay. So why does the bacterium sometimes catabolize the carbohydrate source using aerobic respiration and other times ferment it, even though, as I said, oxygen is present? right? In our lab atmosphere, right, there is oxygen. This is referred to as the Crabtree effect, where they do fermentation instead of aerobic respiration. So, like Bacillus subtilis, right, for example, according to the chart of characteristics for the organism used in our course, right, it is an obligate aerobe, which means for the FTM tube, it stayed at the top, right? Yours stayed at the top. And for catalase, though, it was positive, which means it has an electron transport chain, right? Why isn't it using it in the case of our phenol red broth? Our phenol red broth shows fermentation, right? You had acid and no gas production, but acid indicates fermentation, right? It fermented that glucose. Then also, it was positive for Proskauer. It fermented glucose and produced acetonin that could be then converted into 2,3-butanol alcohol. So what's going on here? There's oxygen in this lab, right? But yet, under these circumstances, right, in these tests, it's actually fermenting. Why is it fermenting the sugar when there's oxygen available? That's the key. High concentration. So when it has a lot of sugar, it's actually faster to ferment it than it is to aerobically respire using the electron transport chain in the Krebs cycle. So Peter made up some numbers, right, to help us understand. But the key to this is that it's quicker. If you have a lot of sugar, it's actually quicker to break it down via fermentation than it is through the electron transport chain. So if it's quicker, even though it generates less energy, but if you can do it faster, you could actually generate more energy, right? So for example, suppose you could get 30 ATP from every glucose metabolized aerobically, right? We talked about this last time. Suppose it takes 1/10 of a second to do that. So within a second, they could produce 300 ATPs. Now suppose there's an abundance of glucose, right? They could sense that there's tons of glucose in the environment. They're only going to get 2 ATP for every round of glycolysis that they do, but they can do it in 0.005 seconds. This is 20 times faster than utilizing the electron transport chain. So they can generate per second 400 ATP as opposed to 300 utilizing Krebs cycle and electron transport. These are made up numbers though, right? We don't know what the actual numbers are, but it explains why, right? If they're faster at it, if we've got plenty of glucose, we don't have to be efficient, right? We don't have to get as much energy as we absolutely need from it. We just need energy fast. And especially for microbes where being able to grow quickly is going to be advantageous, it makes sense to be faster than be efficient. Make sense? Yeah, it's going to use more glucose, right? Because you need more glucose to generate more energy. So in this case, right, they're getting 30 ATPs for every molecule of glucose, where in glycolysis they're only getting 2. But if you have an abundance of glucose, then who cares? Just use a whole bunch of it really fast and generate a whole bunch of energy and grow really fast compared to the other organisms that may not be able to do that. Make sense? Although slow and steady usually wins the overall race, right? The turtle in the hair, yeah. So this is referred to as enzymatic kinetics, right? There are enzymes that catalyze these reactions that determine how fast they're going to go. So there are some different enzymes out there, they've calculated like how fast these enzymes can go. So notice some of these are really fast. I mean, generating their 600,000 of their products per second, right? Others are a little bit slower, right, because of what's involved in the process, like DNA polymerase, right, generates 15 nucleotides per second, but that's still pretty fast, right, considering what's involved in the process. The enzyme lysozyme that can destroy peptidoglycan, right? Only destroys about half of the molecule in a second, right? It's not very fast, right? But it eventually will get the job done, right? And it doesn't necessarily need to be fast. Other, right, that are really highly necessary, right, work very fast. This also, you have to take into consideration how much of the products are substrates to make the products are available, what temperature, right, what pH these reactions are happening at. I noticed in this table it doesn't specify the specifics of those types of conditions, but environmental conditions also affect the speed of enzymes. If you think enzymes are super cool, right, this is our plug for our science laboratory technology program, commonly referred to as biotech. So organic chemistry and biochemistry obviously goes into, right, these types of things. And in the biotech program, protein expression and analysis deals with this kind of stuff, and how do proteins work. Most of your enzymes are proteins, right? And these are catalyzing the reactions that run our cells. So this is a brand new approved program at Delgado. So if you like lab and the hands-on stuff, and that may be something you're interested in, they're having an open house. I'm going to blast the information to all my Canvas classes, so you guys will see an announcement from me. But if you've had contact with Clint Coleman or Brent Fedora or James Gunther, they're the three main guys that run that program. I also help out in the summers with the offshoot of the program for kids, the STEM, the summers camp program that we do for middle school and high school students. And we're going to have an open house. As I said, I'll blast this to you guys. November 19th is a Thursday night from 5 to 7. I'll be here, as well as the guys will be here. And the lab that they're setting up, which is across the hall in 100W, that's where we'll be at. So if you're interested or you have questions, right, there'll be light refreshments. If you're around on Thursday evenings, come on by, right, and find out, check out the lab. Lots of cool opportunities if you like this kind of stuff. The biotech industry is getting huge now, so lots of jobs out there. This is different from the medical lab tech program we have here at Delgado. That's specifically for, medical processing of patient samples, right? Where biotech is a little bit different. Looks into a lot more stuff. So for this lab, we're looking at 3 specialized media, plate media, that you guys inoculated. So all 3 of these media have these 2 characteristics. They're selective media, which means they favor the growth of some microorganisms and inhibit the growth of others. So there's usually something in it that's toxic to the organisms we don't want growing. So that's the selective agent we're looking for. They're all also differential, which means that we'll be able to distinguish different groups of organisms depending on how they grow on the plates, their biochemical characteristics. What can they do? And one of the common questions we've been asking lately is fermentation, right? Can they ferment sugars? So for 2 of the plates that we're looking at, we're going to be looking at just that. We're going to be looking at fermentation. The other plate, we're going to be looking at their ability to destroy red blood cells. So for the mannitol salt, which is a selective media, for which group of organisms are we selecting? Yeah. The ones that we're typically looking for are going to be gram-positive, but a little bit more specific. Staphylococci. And in truth, that's true for clinical specimens, but the main selective agent in this media is a high salt concentration. So not just the salt, but that it's a higher than normal concentration of salt. So salt-tolerant organisms are what are going to grow on these plates. Typically, when we're looking at clinical specimens, we're looking for Staphylococci. These are very salt-tolerant genus of bacteria. But some of you guys that have some bacilli, they're not human pathogens. They're things that live in the soil. that also happen to be salt tolerant. So you may have some growth on these plates, indicating that it's salt tolerant, not that it's a staphylococci. Does that make sense to you guys? Right? So really what it tells you that it's tolerant to salt. And again, it's 7.5% salt. This is a very high concentration. To give you an idea of isotonic saline, right, balanced for our bodies, is 0.9% salt. Okay, so this is very high. So the plates look kind of pinkish. What compound in the media makes this media differential? Nope. Phenol red is a pH indicator. That's just going to tell us pH. Mannitol, right? Mannitol is what's going to be fermented, right, by the organisms. That's the differential component. Without the mannitol, they don't have anything to ferment, nothing to change the pH. So because we are looking at something that can change a pH, fermentation of a sugar that results in the production of acid, right? So mannitol is this sugar alcohol which can be converted into fermentable sugar, right? Pyruvate, right? And be fermented, produce gases, alcohols, and acids. So in order to detect the pH change, what is the pH indicator in this media? This is phenol red. And this is just like the phenol red test we did, right? The broth tests we did. So we know the color changes for this one, don't we? So when acid is generated, what color is it going to turn? Yellow. Right? So I have a demo. Notice one section of this Mannitol salt plate is yellow. Guess what I put there? I put an organism that's salt tolerant, right? That ferments the mannitol, therefore produce acid and turn the phenol red yellow. So what common organism does this that we look for in clinical specimens?
Aureus.
You have a staph. So Staphylococcus aureus is the one they're typically looking for when they're running this test. For this, they'd say to also put the bacteria on Nutrient Agar. We didn't do this because you guys have been doing this all along, right? On Nutrient Agar or TSA, general purpose media that's not selective or differential, right? It allows the growth of just about any organism. Why would you do this, though? To compare, right? Act as a control. So, again, we didn't do it because we've basically been doing this all along, right? You had it growing on normal media. In a clinical lab, they wouldn't necessarily do this either because, again, they don't care about the other organisms, right? So, for instance, if you streaked it onto your plate and then onto this plate and it didn't grow, they're like, oh, well, we don't care that it didn't grow. It means that it's not what we're looking for, right? For you, it would let you know that, okay, my organism can grow. It just didn't grow under these conditions. It's not salt tolerant. So what would be the likely consequence of omitting the salt? So if there's no salt, yeah, nothing's being selected for, nothing's being killed, right? This media would no longer be selective, right? We no longer will be favoring the growth for salt tolerance. We wouldn't know what was growing on it. We may even be misled, right, if somebody screwed up and think maybe it was a Staphylococcus when it was not, because it was able to grow because there was no salt. So that would give you potential false positive results, right? We definitely don't want that. So as we said, this would affect specificity or sensitivity? Specificity. How specific this test is for salt-tolerant organisms. If you don't have the salt, You can't be specific as to what you're looking for. So as we talked about with the citrate test, which was a really mean test, right? We gave them 1 carbon and 1 nitrogen. All food sources for microorganisms, and including ourselves, must contain carbon and nitrogen. If you don't give them that, they can't grow. In this media, you'll notice that for carbon source and nitrogen source, beef extract, this is what we call a complex media, will provide all the nutrients necessary. But in addition, peptone, pre-digested protein is given. This helps them grow faster. It's like giving them baby food. And then as far as carbon is concerned, you have the addition of that sugar alcohol, mannitol. So this really helps them and is most commonly, of course, used as an energy source. And again, there's so much of it, they actually choose to ferment it, even though they can respire aerobically. And so they get the acid production. And carbohydrates yield more energy than protein, so that's what they're going to use as their energy source. With the diversity of microorganisms in the world, can a single test such as the Mannitol Salt Agar be used to confidently identify Staphylococcus aureus? Could I give you a sample and say, oh, here, just use mannitol salt, and if it comes out yellow like that, you know it's Staph aureus, right? No. As I said earlier, right, some of the bacilli will grow on this and even over time ferment. But in a clinical setting, chances are it's going to be Staph aureus, right? But if you're using samples that you don't know where they came from, they didn't come from a person, right? They could be an environmental sample, then it's not going to be very reliable. And again, even in a clinical lab, this is a starting point. They would do additional testing, right? So it's not the be-all, end-all, right? It's a presumptive test. We can presume, okay, let's move forward. We probably have Staph aureus. So for this plate, what do you see? Do you see growth?
No.
Yeah, that's growth. That's growth, right? On that plate in the non-yellow section, one of those other sections, do you see growth? Yeah, look at that.
Oh.
There's some growth there. But it's not yellow, is it?
No.
No. So it's very similar to this picture. So what could you tell me about this organism?
It's not Staphylococcus aureus?
It's probably not Staph aureus because it would have turned yellow.
And it can't survive in high salt content?
It can't. It grew. So it can, right?
Yeah.
It can grow in a high salt concentration. But it's probably not Staph aureus. And in fact, it's Staph epi. Epidermidis. Right? And that's what's on those plates as well. It'll grow in a high salt environment. It grows on our skin. That's very salty sometimes, right? But it does not ferment the mannitol. Right? It's a non-pathogenic staphylococci. Very common to our skin. Make sense? Okay. So, what would we see if it was Staph aureus?
Yellow.
It would be yellow, right? It's not yellow. So, it didn't ferment the mannitol. It's probably not Staph aureus.
So this was originally this color. It turned yellow.
Yes. Yes. So, part of the reason why I gave you guys the plates the way I did, you've got Staph aureus and Staph epi side by side, and the other section I didn't inoculate, right? That's the color, remember, the plate started out, this pinkish or orangish color, right? So in this case, right, the acid was produced and it changed color to yellow. So again, side by side, you can see growth on both sides, but the difference here for Staph aureus is that yellow color because of the acid production and that pH indicator phenol red telling us that it is acid by turning yellow. So note this plate, although not a great picture, if you look closely, the organism itself is yellow. We're not looking for that. We're looking if the media has turned yellow. And in this case, part of it's orange and part of it's pink, right? This is one of the bacilli. As I said, they will sometimes grow on these plates, but that's just indicating they're salt tolerant, not that it's a Staph aureus. And sometimes, if given enough time, it will actually turn it yellow as well, which means it's doing what? Fermenting the sugar, okay? But it does not mean it's Staph aureus, right? So it'll help us isolate what's called halophilic, referring to salt requiring, or haloduric, which is sometimes referred to as salt tolerant organisms. Typically this is Staphylococci, but as I said, some of the bacilli also are salt tolerant. Among the survivors on the selective media, the ones that are salt tolerant, then we can ask the question, can it ferment mannitol? Can we ask that question if your organism didn't even grow on these plates? No, right? We don't know if it ferments mannitol because it didn't grow. So for some of you guys, it won't be determined, right? Because they didn't even grow to begin with. Make sense? So we just spot inoculated, right? Put some bacteria on the plate, right? So that you can come back and see it. I've got them stashed in incubator, refrigerator all over the place. I will take them out, as I said, and go over with you guys your results individually. So media surrounding the colonies remains red, means no fermentation happened, right? If it's the same color as the uninoculated section. The media surrounding the colony, if it turns yellow, we know acid production happened. We know fermentation happened of that mannitol sugar. So growth, the organism is salt tolerant. That's what growth tells us, right? And it's only the color change that lets us know if it fermented the mannitol and if it could possibly be Staph aureus. But of course, you guys ran other tests, right? So at this point, you know whether you have a bacillus or a staph, at least, right? Maybe trying to figure out which staph. So that plate will help. So the next plates that we're going to look at is the Columbia CNA plates. And these contain 5% sheep whole red blood cells from sheep. These are very similar to human red blood cells, and so that's why they're utilized in this test. They are intact because what we're looking for in this test is whether the organism can produce a set of enzymes known as hemolysins. These literally lyse, these are toxins that they produce, exotoxins that leave the bacteria and attack the red blood cell membranes. So, why do some organisms produce these? What element are they after inside the red blood cells? It's an element. So, carbon, hydrogen, right? What element associated with hemoglobin? Iron. The carries the oxygen, but they're after the iron. They need it for their metabolism as well. Iron is very precious to us and microorganisms. So as you know, our red blood cells, the hemoglobin, that protein component, the iron, that element component of the hemoglobin protein is actually what interacts with the oxygen and allows us to carry it efficiently through the body. So that's a rich source. So if they can get at it, right, that's very beneficial to them. So having these exoenzymes that can lyse the membrane of our red blood cells is very beneficial to them. So this media is selective. And it's selected for a particular group of organisms. In this case, it's gram-positive or gram-negative. It's gram-positive. And the reason for that is the selective components are what?
Colistin.
Yeah, two antibiotics, colistin and nalidixic acid. And again, I won't make you spell these, but you do need to recognize them. So notice colistin, however it's pronounced, has very similar, right, to E. coli, Escherichia coli, which is a gram-negative bacteria. So it's effective against gram-negatives, so they don't grow. It's only gram-positives that grow. And again, because this characteristic is very common in gram-positives, and we're typically looking for pathogenic gram-positives with this ability. Not to say that gram-negatives, some of them don't have this ability. But for this particular media, we're only going to see gram-positives, which is why, again, I only gave it to you guys if you had a gram-positive organism, right? We'd be wasting a plate if you had a gram-negative because the antibiotics would have killed it. No growth would have happened. And we have to buy these plates, so we don't make them. I don't want to mess with red blood cells from sheep. No, thank you. So this is also differential. So we're selecting for gram-positives by the antibiotics killing off the gram-negatives. Then now we get to the differential component. What in those plates is going to get changed by these organisms? The red blood cells, right? Those red blood cells, those whole red blood cells, is what's necessary. And that's why it's bright red, those plates. And then sometimes in my notes, it autocorrects and says can instead of CNA. Drives me crazy. I think I've caught them all, but, some may have escaped. But it's C for colistin, Na for nalidixic acid. And so you'll notice, as I said, we purchased these from a provider, so you'll notice they're stamped with a date and with CNA with their 5% sheep's blood. Columbia is the base media. So there's other food in there, too, for them to grow. So there's two different types of lysis that can happen. What we refer to as beta, and this is complete destruction of the red blood cells. So when I give you guys these plates, I'm going to ask you to hold them up to the light, and especially for one of them, it completely clears it. You can see right through the plate. The red blood cells have been completely destroyed. The other type is alpha, and this is incomplete. It just pokes holes. And so oxygen gets in, and under these circumstances, it turns kind of greenish, almost like rusting, right? Because remember, iron, when exposed to oxygen, rusts. So how I remember these two is I always think B, beta for bad. Like this is the worst case. It completely annihilates your red blood cells. So the causative agent of strep throat, Streptococcus pyogenes, or pyro, I forget how that one's pronounced, does this. So when I was a kid, you go into a doctor's office and they thought you had it, they would swab the back of your throat and literally right there in the doctor's office rub that onto a blood agar plate and stick it in the incubator. Right? They don't do that kind of stuff in the office anymore, right? They swab, put it in a tube, and send it off to the lab. And somebody in the lab does that. But they also can run a quick test, similar to a test that we're going to do the last lab, right, in 2 weeks, called the ELISA, where they're utilizing antibodies attached to an enzyme to create a reaction. If the antibody binds, then an enzymatic reaction happens. It gives a color change, right? And so you guys are going to do that in the lab, right? We're going to play with antibodies and the enzyme and create color change if a particular substance is there that the antibody is recognizing and binding to. So in this case, if they get enough bacteria in the swab and enough of the antibodies bind, a color change happens and gives a positive result. So they very rapidly, literally within minutes, can determine whether you have strep throat. What's the benefit of that as opposed to swabbing it onto a plate? How long does it take before we read results when we're looking at plates? You're going to have to wait till at least the next day, right? 12 to 24 hours, right? They usually would call you, right? Take the antibiotics I prescribed. You have it, right? Now they could tell you right then in the doctor's office, potentially, right? Depending on whether they get a good enough sample. They're still going to send the swab off to the lab to confirm. Make sense? Because you can get a false negative reading from this test. But it helps us in treating faster. Right? Diagnosing faster, treating faster. And if you've ever had strep throat, yeah, you know. you want to feel better faster. So this is alpha. This one is the incomplete lysis. And so again, the greenish discolorization you will see, especially when you hold it up to the light. What's this one? This one's beta. So notice the clearing around the growth, right? This is Staph aureus. This one also tends to be beta hemolytic. It is a gram positive, right? So it'll grow on these plates. So we have beta, and of course it's gram positive, because remember, gram-negatives won't grow on this media. Gamma hemolysis is actually no destruction of the red blood cells. There are organisms that are gram-positives that will grow on these plates, but they won't cause any destruction to the red blood cells. So this is an example of that. This organism that this student had that semester, notice it's growing on the blood agar, right? It's growing, but they had a contaminant. And this is a very common contaminant that grows in our nose. It was probably Staph aureus, probably breathed on this plate. So this is a contaminant, right? There's just a little bit right here. But in this case, what type of hemolysis are we seeing?
Alpha?
Beta. Beta. It's completely lysing. See how it's clear? Big clear zone? Right? So again, it's probably came from our own nose, right? Most of us carry it at one time or another in our noses, right? Just breathe in our plate. Remember, cover them up with our lid. That's why we do it. But that was a great example for my lectures. So again, notice growth. When you hold it up to the light, you see this clearing, right? That's beta. This one is the two staph side by side. Notice, again, Staph aureus. Notice the clearing, right? And then no change in the blood agar for the staph. Actually, I think we have descriptions here, yep. And then this one is similar to what I'm going to hand out to you guys today. We have the alpha, see the greenish. The beta, you see the clearing. There's actually a couple zones of clearing. This one is another beta that really clears out the plate. I mean, you can literally see through it. It's destroyed all the red blood cells. And then it's a bacillus. So as it runs out of nutrients, it forms spores, so it almost seems like it goes away. The growth over time goes away. And then this one is, it's growing, right, but there's no change to the red blood cells. So that's what we call gamma. So again, beta, gamma, and then again, notice growth, right? So it's probably some gram-positive, right, but no lysis of the red blood cells. So I have two demos, so pass them around. That again, when you hold them up to the light, right, you can see clearing, definitely for this one, right? And then the one that's labeled SA, you should see some clearing around that one as well. Not as definitive as the bacillus one, because again, as the bacillus dies. The M one that's greenish, that was from my mouth. We don't actually have any really good alpha, and most of us have Streptococcus mutans in our mouths, so I swab my mouth and put it on this plate. So we'd have a nice green one. It's too hard to grow those guys in the lab, right? But we carry them around with us all the time. EF is Enterococcus faecalis, and some texts say that that one is alpha. It doesn't look very green to me. But definitely you can really see that beta. So if you guys on this side of the room, pass this around, hold it up to the light so you guys can see different degrees of hemolysis. So additional testing that's done on especially streptococci, which again we don't deal with in this lab. A lot of them are pathogenic and hard to keep happy. You'd have to keep growing them on blood agar, which would be expensive. We run serological tests where they're using antibodies, and they're looking for polysaccharide antigens, so these sugar antigens that are on the surface of streptococci. And so there's lots of different Landsfield groups. Many, but not all, are beta hemolytic of these streptococci. This is a different test, right? So we're looking for and distinguishing the different ones. So there's numerous groups. Group A of the Landsfield test are most of the pathogenic ones, like Streptococcus pyogenes, which is the one that causes strep throat, right? So this is the one that we're typically looking for. Streptococcus galactate falls under the group B, which causes infection for cows, mastitis, infection of their mammary glands, right? It can cause serious problems. And it can be found in normal milk. It includes strains that are even found in our own throat and vagina. The beta strep tests that they're typically doing on women before you give birth, they're testing again for these types of organisms that may be present in the vagina, because they can be part of our normal flora that don't cause problems for us, but when the baby comes out, it passes right through the vagina, right? Through the birth canal or the vagina. It's the same thing. And so they could get exposed to these streptococci and cause serious problems for them. So they always test, and they're going to give you antibiotics if you have it, so that hopefully you don't pass it on to your child. So as I said, the different groups, some of them are beta hemolytic. A lot of the ones in the B group, right, this one that we're looking for in the beta strep test, that's what they're looking for as this organism. So these Staphylococci, right, have a carbohydrate associated with their cell wall, their outer component of their cells. And that's what they're testing for, and that's how they put them in the different groups. So different groups have different carbohydrates, right, and give us an idea of the different type of streptococci that may be present. So again, why don't we do the Nutrient Agar plates? We really don't need to, right? But it could be done as a control. So early formulations of this media had a slightly higher concentration of colistin and nalodistic acid. Remember, these are antibiotics, right? How do they work, right? So colistin is an antibiotic that affects membrane integrity. And nalodistic acid interferes with DNA replication. So it interferes with the enzyme associated with DNA replication. So 4 gram negatives, it affects their outer membrane. Acts as a detergent similar to ethanol and the gram staining, so it disrupts their outer membrane. But at a higher concentration, it could potentially disrupt gram positives. So that's why they had to make sure they had the concentration just right in these plates. Otherwise, we could also affect the gram positives, and we don't want to do that, right? We're selecting for them. We want them to grow. Same is true. It's a different enzyme for gram positives versus gram negatives. For this that interferes with DNA replication, of course, all cells do that, but different types of cells use different enzymes. And so the enzyme that they're targeting is used by gram negatives and not used by gram positives. But again, anything in a higher enough concentration can be disruptive. So we had to make sure we get the concentrations just right. So colistin, again, remember this outer membrane is what's being attacked for these gram negatives, and why they're so much more sensitive to particularly this membrane integrity disruption antibiotic. So why are we doing this? Well, we want to isolate gram positives. So again, only if yours is a gram positive is going to grow on these plates. Especially when we're doing this, we're typically looking for these types of organisms: Staphylococci, streptococci, and enterococci. But other gram positives, right, we're using are not clinical specimen type stuff, so they will grow as well. Among the survivors, then we can determine, right, can they then disrupt red blood cells? Can they completely disrupt what we call beta hemolysis, or just lightly? So we tried to do some streak plates. They didn't come out so good. But again, we wanted to inoculate the media so that we can then look at our results. So we can determine if we have hemolysis. If so, what level? Is it complete, beta, incomplete, or no lysis at all? If we have growth, again, it's a gram positive. We do know that. If we have clearing, it's beta. If we have partial lysis, that greenish discolorization, it's probably alpha. No alteration of the media or colony, right, is what we call gamma. No hemolysis. Of course, you can't tell hemolysis if it doesn't grow at all, right? And again, those of you guys that weren't going to grow, I didn't even give it to you, right, because you had gram negatives. The antibiotics were going to kill it. So the last media, this one's my favorite, eosin methylene blue with lactose. In your book, it mentions sucrose. But that sugar isn't in all the formulas of EMB. And the ESN methylene blue, that the formula that we buy, only contains lactose, because that's really all we were testing for. We want to know if it can ferment lactose. So the selective component, though, what stops the gram positives in this case from growing? It's in the name of the media. Yeah, the two dyes, eosin and methylene blue. So gram negatives grow. The selective components are eosin, Y, and methylene blue. The two dyes that are in the media are actually toxic to the gram positives. So if you had a gram positive, it shouldn't have grown on these plates. So what makes it differential? Lactose, right? Lactose is the sugar that's going to be fermented. So as I said, in your book it says and sucrose, but we use the formula that only contains lactose. So it's important to know which one you're using, right, if you utilize this media. So this media is particularly used for coliforms or fecal coliforms. So these is a general broad class of organisms and not a specific classification. But they're for ones found in the environment, but also in feces, right? So they let us know potentially if certain things have been contaminated by fecal matter, if we can detect the presence of these types of organisms. These organisms are lactose fermenters, so hence why there's lactose in this media. They typically are gram-negative bacilli. So again, we're selecting for gram-negatives. We don't want gram-positives growing. We want to know, do we have gram-negative lactose fermenters? Do we have a possel coliform, or even worse, fecal coliform, one that came from fecal matter? So they're defined again as enteric. So they're typically ones that come from the gut of warm-blooded mammals. Again, ferment lactose, produce acid and gas. So the presence of coliform bacteria, right? These lactose fermenters can indicate that drinking water may have been possibly contaminated, right, by fecal matter and be possibly harmful. You might have disease-causing organisms. So approximately 90% of fecal coliforms are Escherichia coli usually. Other genera of bacteria that will be found in feces are Klebsiella and Citrobacter. And as we know, Klebsiella can be pathogenic, right, like Klebsiella pneumoniae. When the pressure in New Orleans drops, right, in our water purification systems to 15 psi, pounds per square inch, or below, the city issues a precautionary boil water. We've been experienced this several times, right? In order for, and this is for 24 hours, because guess what they're going to do? They're going to test it. They're going to test it by running cultures, right? They're going to do things like this, where they're going to filter the water through a filter. The membrane is going to trap the bacteria. They're going to put it onto a plate and see if anything grows. We obviously do not want fecal coliforms to grow, right? They'll either put it on EMB or McConkey agar, that filter, right? And then we'll look for growth. Here you can see growth has occurred. This is not a good sample, right? This is an example of contamination. Thankfully, what they consider potable water, right, has less than 1 coliform per 100 milliliters of sample, right? This is definitely more than 1, right, in the picture. We don't want that. So we've had this happen to us several times over the last 5 years. Seems like more, but I'm sure Peter got accurate data. And each time, no contamination has happened. So that's the good news, right? The bad news is that we have to be cautious and fearful that it may have gotten contaminated because they're not got the best system for maintaining water pressure. So hopefully they will fix that. So foods can become contaminated from a variety of sources, right? Gram-negative coliforms, gram-negative non-coliforms, and gram-positive bacteria that can cause problems. Several come either from the soil or water, right? Others are enteric ones that come from the guts of humans and animals. Note that coliforms are found in the environment as well as in the gut, right? So you do have environmental sources, although fecal coliforms are found only in the gut, right? So when we're talking about coliforms that are fecal, they're ones that are only found in the gut. They're the enteric microorganisms. So, right, so here's ones found only in our gut. Here are coliforms, right, in general that can be found in the environment. Notice some of these guys can also be found in the gut. E. coli is the only unique one, right? It's not found in the environment unless you have fecal contamination. So it's one of the big ones that we look for, right? And it itself usually isn't pathogenic for us, but the fact that it's there means that feces probably got into that sample, right? Because it's a gut-only fecal coliform, right? So it means that maybe other bad guys are present in that sample, whether it be a water sample or a food sample or anything like that. So people freak out about E. coli, although E. coli itself usually isn't very pathogenic unless it's a pathogenic strain. It's all the other ones that potentially are there because that one's there. Because the only way that one's getting there is fecal contamination. Make sense? Okay. So Enterobacteriaceae, this family, right? Your primary pathogens. And then again, as I said, this is the only one that crosses over E. coli. And others are opportunistic pathogens, right, when the opportunity arises. So again, E. coli can be bad news when it gets places it's not supposed to go, right, outside of our gut. So how does food get contaminated? Peter picked this picture, I did not. Right, unsanitary slaughter and processing practices, right? One of the main reasons why some people don't eat animals other than the fact that they don't like this kind of thing happening to animals. But it's how it's done that matters, right? How it's processed. Water and soil can be contaminated and therefore contaminate crops. Direct contamination by the people handling or preparing food, right? Food handlers, poor sanitation practices, right, are one ways that they can contaminate food as well as ourselves. Staphylococcus aureus, which is a gram-positive bacteria, right, we've been learning about. Some strains can produce an enterotoxin, so this is a toxin that affects our gut. And so therefore, if you ingest food contaminated with this particular strain, it can cause a particular type of food poisoning. So notice again, it's gram-positive, so we're not going to detect this one when we're doing EMB testing for coliforms, right? Topidine poisoning was specifically given the name for Staphylococcus aureus orientation of food poisoning. And there's a aside as to how it was given that particular name. Typhoid Mary is my favorite food handling mishap. Her name was actually Mallory Malone, and she was an immigrant from Ireland. And she worked as a cook. And after several people got typhoid fever, after ingesting her food, they were able to link it back to her, that she was the source of the contamination. Herself was not sick. She had been sick previously, and unfortunately for this particular salmonella bacteria, it can colonize the gallbladder. And so each time bile is excreted into the digestive tract to help with digestion of fats, it goes into the feces. So number one problem here is she's a carrier. Number two, clearly does not know proper sanitation, right? Wiping her hands, cleaning them properly after going poo, right? Okay? And is exposing others. Back in that time, they didn't have antibiotics. They also probably didn't understand this carrier state situation. So they quarantined her. Back then, y'all, they jailed her, right? Told her she could no longer be a cook after several years and released her. That's the fourth problem. Did they train her to do anything else to make a living? No. So what is she going to do? Go back to what she was doing before. Not to mention training her how to wash her hands properly. after going to the bathroom. So she goes and becomes a cook in a maternity ward, and guess what? A whole bunch of people get sick. And it gets linked back to her. They quarantine, imprison her again until her death. When they autopsy her, they find out that her gallbladder, right, was infected with this particular bacteria. There are several ways we could have solved this problem and failed. Education is key, right, to success in this world. Just being in this classroom, right, benefits you and everyone else, hopefully around you, because you won't make these mistakes. Yes? Okay. So, Salmonella and Shigella. There are, of course, special procedures for detecting the presence of these types of organisms, special media to be able to detect the presence of these pathogenic microbes that can cause serious problems that are coliforms. They could come from feces, but they can also come from the environment. In turtles is the big one. You got to look out for Salmonella, in addition to what everybody knows about chicken, right? But in Louisiana, right, don't forget about good old turtles. Wash your hands after playing with them. All right, so E. coli, as we said, is a fecal coliform. This one, the reason why it's my favorite is this discolorization that happens to it as it grows on EMB. As it produces a large amount of acids, because it's a very strong lactose fermenter. Which clock is right? Okay. When me panicked. Okay. Like, I think I was that far behind. Okay. So this metallic green, it turns black, and then this pretty metallic green sheen. So this one's my absolute favorite. Again, I got 2 plates for this one, so pass them around. And this one, you definitely, I got some tape on it, but just so that it can be hinged. You want to go ahead and open this one up because it gets kind of foggy and you can't see. But you can clearly see the E. coli on this plate. So one for this side of the room and one for this side of the room. So go ahead and take a look at that. Notice that pretty metallic green and pass it on. So again, it's not detecting the gas, but just the acid. And it's the interacting with those dyes, the eosin and the methylene blue, is what gives it this pretty color change. If it produces a small amount of acid, the color change isn't quite dramatic or as beautiful as that one. You're going to see pink or purple growth, right? So you'll notice the other 2 swabs on there are kind of pinkish and purplish. They're fermenters, but not to a great degree. So here, right, this one that's almost blending in with the plate, that's a gram-negative, but it's not a fermenter. Where the purple you see here, that's fermenting lactose, and this metallic green is definitely, right, fermenting. And for our lab, that is E. coli that does that. It's the only one that does that. So again, why would we do Nutrient Agar? It would be a control. We didn't do that. We've been doing that. You know what your organism looks like. So dipotassium phosphate is a buffer that's added to this media because of the production of acid, right? It helps keep the growth and so that the acids don't kill the organism. In the environment, they would be able to get away from their toxic byproducts, right? So the buffer helps with this growth. But also, it affects pH, right? So what would be a possible consequence of adding buffers to raise the pH to 7.8? Well, think about that. That's a little bit above neutral, right? Instead of at neutral. So what are they going to have to do to get the pH to go acidic? Work harder, produce more acid, right? And because of that, we might not be able to tell the difference, too, between E. coli, that metallic green, that really strong fermentation, and the lighter purple-pink fermentation, right? E. coli might come out looking purple or pink, where normally it would have its metallic green sheen if the pH isn't adjusted properly in these plates, right? And would give us some false readings. So would this affect the media's sensitivity or specificity? Sensitivity, right? We wouldn't be able to discern the different results. So again, there's lots of organisms out there. So if it's a metallic green sheen, can you confidently say E. coli?
No.
No. In this lab, yes, right? It's the only one we've got. But, you know, there's no definitive. But when in Antarctica, right, if it looks like a penguin, it probably is a penguin. If it looks like E. coli, it probably is E. coli, right? All right, so here are just a few more pictures, right? So notice this one, you don't see those purple or pinks or the black or metallic, right? So what can you tell me about this organism? It's gram-negative, right? Because gram-positives are killed by the dyes. But it's not a lactose fermenter, right? So it is gram-negative, but it is not a lactose fermenter. This one is pink, right? So is it fermenting? Yes. Notice this one, again, about the same color as the media. So any fermentation? No. No fermentation. We get some really cool different growth patterns on these plates. As I can say, right? See these pinkish purple ones? That's fermentation. This one probably isn't. Although I see a little bit of pinkish to this one. One of the organisms that we work with, so these are all fermenting, right? We all see color changes. This one is Klebsiella pneumoniae, which if you really compare this one to growth on regular Nutrient Agar, it's shiny. On eosin methylene blue, it gets this dry, cracked appearance, rough looking. Its growth pattern really significantly changes by the growth on this media. Why, I don't know, but it's really cool to see. So that one's a pretty good dead giveaway. And then, of course, the E. coli, as I said, that fluorescent green sheen. That's really pretty. So why do we do this? We want to isolate, right, identify gram-negative. So if you had a gram-positive, it shouldn't have grown on these plates. The dyes will kill it. If it survives, we then can ask the question, can it ferment lactose, right? So we spot inoculated, right? Colorless colonies, no fermentation, probably not a coliform. Pink, modest fermentation, maybe a coliform. Purple or black or sometimes metallic green, very robust fermentation, usually a coliform. One of the major ones that does this is E. coli. So growth, again, means gram-negative, and then the degrees of color change determine the amount of fermentation that happens. Trying to click there and it's not letting me click.